today I'm going to talk to you about genomic registries and kidney research. And these are my disclosures. And so I wanted to get started to tell you, so why are we doing all this research, or why as clinicians, what do we do? Because we're trying to answer a number of patient questions. The patients have questions for us. Typically, they want to come to us to find out what is this disease that they have. And they want us to know, why do I have the disease? What will happen to me? And what are the treatment options that are available? And all of us as clinicians and scientists are trying to answer these questions to the best of our knowledge and capabilities. A new question that has arisen is that we have the ability to perform genome sequencing. And now a lot of genome sequencing is actually available as direct to consumer products. And so many patients may actually come to us with their genome sequence, or parts of their genome sequence, and they want to get the physicians to interpret this information for them. So every clinician and nephrologist will have to be equipped with some knowledge of genomics to be able to answer some of these questions, particularly regarding interpretation of common and rare genetic variation and how those may relate to disease. So how common are genetic diseases of the kidney? Well, it turns out that if you look on the left, this was before the really systematic sequencing of large cohorts with CKD. There was a review that was performed in the Lancet by a group of investigators led by Olivier de Vust, who looked at the literature and came with an estimate that generally about 10% of patients with chronic kidney disease, pediatric and adult, may have some form of inherited nephropathy, and often those may not be recognized. Our group started to investigate this a little bit more systematically when we wound up going with the exome sequencing, which is the sequencing of the coding portion of the genome. And this technology works very well for detection of essentially Mendelian or monogenic forms of disease. We wind up sequencing about 3,300 patients with different forms of CKD. And altogether, we identified diagnostic mutations in about 9.3% of the cases. So these are patients who had Mendelian forms of kidney disease. And so in this slide, you see there were 307 patients that had a genetic diagnosis of a Mendelian form of kidney disease. And there were 66 different distinct genetic diagnoses that were made in this cohort. But these were not sort of distributed equally across these 307 patients. There were 6 genes which accounted for nearly 2/3 of the diagnoses. These were genes with mutations in PKD1 and 2, diagnostic of polycystic kidney disease. Type 4 collagen mutations, typically associated with Alport syndrome with thin basement membrane disease, and neuromodulin associated with autosomal dominant tuberostitial disease. And then another 1/3 of patients had mutations in 60 different genes. And so these patients essentially were all Ns of 1s, really rare diseases. And for the most part, they were not recognized and diagnosed in the clinic based on the standard clinical evaluation. Another way to look at this was looking at the diagnostic rate based on the clinical categorization. So what is the clinical diagnosis that the patient had when they came into the study? And you could see that on the y-axis, you can see the diagnostic yield. So to the right, you can see the diagnostic rate for Mendelian disorders was 1 to 2% if the patients had a diagnosis of hypertensive or diabetic kidney disease. So the misclassification rate was pretty low. On the other hand, the diagnostic rate was nearly a quarter in patients who had a clinical diagnosis of a congenital cystic disease. So your pre-test probability for a positive result really was influenced by the clinical workup and the clinical diagnosis of the patient, and trying to think about then the workup and the diagnostic yield based on your clinical assessment is going to inform your choice of testing and the way you're going to approach this for your patient care. And what was really interesting, so glomerulopathies and tuberostitial diseases had somewhat an intermediate diagnostic yield. And finally, what was really gratifying for us was that 10% of patients had kidney disease of unknown cause based on the clinical assessment, but yet we were able to come up with a diagnosis based on sequencing in essentially 1 in 5, 1 in 6 patients. So this told us that essentially diagnostic testing is going to be really powerful in the management of patients with kidney disease, and is going to have a bright future for clinical practice. And we're not the only ones who've really done this. A lot of different studies have now implemented genome sequencing, and this diagnostic yield has ranged somewhere between 5 to 60%, depending on the way you wind up selecting the patients and the diagnostic modality you use. So this is going to happen in the future for all of us, and we're going to wind up using the genomics for diagnostic testing. So how about for kidney research? What else can we do to enhance more discovery? Because we're only making a diagnosis of monogenic diseases in about 10% of patients. So one tool that we have at our disposal are electronic health records, because they're widely implemented nowadays across the world. They're semi-structured. There are long-term data in many health systems that are available. And so now there are ways of also translating this data and combining these data sets across different health systems and different records. So an example of this is this network called Odyssey, OHDSI, but they pronounce it Odyssey, which combines de-identified EHR data from 52 databases from 4 countries. And there are at least 250 million patient records, which enable clinical research. We can look at the Odyssey as the ultimate chart review, essentially, to be able to really ask some questions about disease treatment patterns, for example. And here, this is an example of a study looking at treatment patterns for diabetes and hypertension, looking at what is the initial therapy for these two drugs across these whole healthcare systems. And in concentric circles, you could see what is the initial treatment for these drugs. So here, for example, in diabetes, you could see that metformin at the time was used in almost 75% of cases as the first-line drug, and then in the other patients, there were other drugs that were used as first-line therapies. And then in the second circles and the third circles, outer circles, you could see what are the second-line drugs that were added to the therapy to enhance and improve glucose control. And so you could see that the treatment pathways for diabetes were reasonably simple. And so when people started on metformin, then they started another drug like a sulfonylurea, and then a third drug. And so you could see that there are individualized almost, or treatment patterns that emerge for diabetes. Now, on the other hand, when you look at hypertension, the first line of treatment, the first drug that's prescribed, really differs widely. And so there's a wide, so it's reflecting the preferences for clinical care. And so there are, thiazide diuretics, diuretics in general, ACE, ARBs, or calcium channel blockers were used almost equally across the board. And then you start to see that there's even more segmentation when you start to look at the second and third line of drugs that were added on. And in fact, what was really remarkable, you could see that in about 10% of diabetes and almost 25% of hypertension patients, they followed the treatment pathway that was completely unique to them across this 250 million patients. So in other words, you could say that we're either doing personalized care and precision medicine for some of these therapies, or you could imagine that we don't have enough guidelines and it's sort of a random draw. So it's probably a mixture of both of these things. So it really gives you an insight about what's going on for treatment patterns. And so the next step, of course, is to say what happens to these patients when they start on a therapy and what are the outcomes that are associated with this. You can do this essentially about clinical trials, which take decades sometimes, or many years to be able to put together and then get an answer for, or you can look again across these large data sets. And so this is an example looking at the same network, looking at the comparison of effectiveness of antihypertensive therapy, the ones that we know about, in terms of cardiovascular and various other outcomes. And here you could see this is a grid looking at thiazides, ACE inhibitors, ARBs, and dihydropyridine calcium channel blockers versus one another, and looking at the outcomes. And so you could see that exactly as the clinical trials had predicted, thiazides are associated with much better outcome for many of these cardiovascular endpoints. And what was, especially comparison to non-dihydropyridine channel blockers, this was very clear that almost all of these drugs worked a lot better. And what was also interesting was that thiazides appeared to work better compared to even ACE inhibitors based on these studies. So I think that these studies can be confirmatory, but also set up interesting hypotheses that one can then confirm in clinical trials. And the good news is that once you have these data sets, you can start to answer and try to ask and answer a number of these questions that would take otherwise a long time to set up and would be very costly to answer in the setting of a clinical trial. What you can do also is try to make a better diagnosis of patients using electronic health records. So here's an example of a study looking for detection of chronic kidney disease using electronic health records, using an algorithm that detects, that looks at all the parameters in the clinical records, such as creatinine, urine protein, and urinalysis, as well as diagnostic codes to put together a CKD staging and a diagnosis of chronic kidney disease. This was done in 1.3 million individuals who were screened at Columbia. This study was led by my colleague Christoph Kurlach. And in looking at these, in developing this algorithm and looking at these health records of 1.3 million individuals, he identified about 240,000 cases of CKD, but remarkably, only about 20% of them had a diagnostic or procedure code that was compatible with CKD. So in other words, 80% of the cases had gone unrecognized apparently in these health records. So indicating an opportunity to be able to implement such electronic health records to identify the patients, to be able to track them better and maybe do early intervention. Another example of doing this is to just estimate disease heritability with electronic health record data. This is again another study looking at large data sets from electronic health records, but using essentially emergency contact information from patients to be able to reconstruct pedigrees. It turns out that when we give emergency contact information, most of the time it's a family member, very often it's a mother. And so by putting together, if two different people give the same emergency contact information, identify them as their mother, then you can reconstruct pedigree across healthcare records. And so they were able to build 595,000 pedigrees and look at the heritability of different diseases, and we're able to compute heritability estimates across 500 different clinical traits. And this is something that was traditionally done, again, in terms of observational studies, painstakingly, like a Framingham heart study, to look at disease heritability. And for some disorders, we never had even heritability. And so as a result of this, now you can estimate heritability for diseases that have never been, which never been measured. For example, I'm interested in IgA nephropathy. You can look for these rare diseases and see how heritable they are. And you can test to see if the heritability of these rare diseases are concordant with other diseases such as IgA nephropathy and HSV, or Crohn's disease, for which there are clinical associations. And then start to think, based on this, biological and treatment hypotheses based on shared heritability between these disorders. Another interesting study looking at, again, the electronic health records and now comparing to the biobank is out of Vanderbilt University, where they said, well, we have all these rare diseases of the kidney, many of them have Mendelian disorders, many of them go undetected. Is there a way we can put together data from the electronic health records to build a risk score for different Mendelian diseases, such as cystic fibrosis or hemochromatosis, to be able to identify the patients who are going undetected? And sure enough, they were able to apply these algorithms and identify a lot of patients who have these rare Mendelian diseases that have gone unrecognized during your clinical workup through these phenotypic risk scores. So you could see that how this is going to help us identify patients with the rare diseases, and then provide genomic sequencing to be able to confirm these diagnoses and provide the right care for these patients. As you can imagine, making a diagnosis of hemochromatosis is actually going to be very important to be able to provide the appropriate treatment earlier on. So you can combine these electronic health records with biosamples. And so these are biobanks that can be developed. And so if you have large registries now of patients that provide both electronic health record data and biosamples, now you can start to genotype or sequence the samples. And because the biobanks are composed of patients with many different conditions, you can select some patients as cases and controls, diabetic kidney disease, IgA nephropathy, and then other people who don't have any of the kidney problems as controls, and perform case control studies across the genome, and many, many different studies. And the way you can do this is simply as the standard GWAS, where you select a patient with a case and control and scan the genome, and that's the genome-wide association study. Or you can flip things around. If there's a specific genetic variant that you're interested in, you want to see what phenotype they're associated with, where you can scan all the phenotypes that are in the records. So you could turn this around and say, okay, what is this SNP? And what are the patients who have this SNP more prone? Are they prone to a specific disease? And that's called a phenome-wide association study. And I'm going to give you an example of this shortly. So these are some examples of biobanks and registries that are ongoing. eMERGE has about 100,000 individuals with GWAS linked to electronic health record data. The UK Biobank has about 500,000 individuals with genomic and EHR data that's freely available to the research community. And there are now hundreds, maybe 1,000 papers that have been published using the UK Biobank data. Biobank Japan has about 200,000 individuals with targeted diseases. I'm involved. I'm one of the PIs of the All of Us program, which is the genomic medicine, precision medicine program in the United States, which aims to recruit 1 million individuals with genomic and electronic health record data. There's the Pakistani genome research. And I'm also involved as an advisor to the MyReason research program, which has been developed by Frenova and Fresenius to essentially recruit 150,000 individuals with chronic kidney disease or ESRD to be able to advance kidney disease research. And that's going to be the only dedicated kidney repository out there. And I'm really looking forward to see this develop so we can use this to advance our knowledge of kidney diseases. So I can give you a quick example of how we can use these types of biobanking data and large studies to get some idea about disease causation and disease associations. So one of the interesting things about patients with chronic kidney disease, particularly in children, is that there's an association with neuropsychiatric diseases. It's known that patients with CKD are associated with all kinds of impaired neurocognition, with intelligence, anxiety, depression, and in adults it's associated with anxiety and depression. And the question is this the CKD itself that's causing the depression, or the other factors involved? And so we started to look at this and get interested in this because we're looking at the genetics of kidney disease in children and subsequently in adults. And we saw that there's a large number of individuals, 8% of children and about 1% of adults, that have a genomic disorders. Genomic disorders are disorders in which there's a large chunk of DNA of a few 100,000 to 1 million base pairs that are deleted or duplicated across the genome. And we saw that we've identified at least 45 different genomic disorders that appear to be enriched in patients with CKD. And these are shown here in these slides. Each one of these green or red dots indicate one of these genomic rearrangements, and they're known associations with kidney, heart, or neuropsychiatric disease. And you could see many of these disorders, because they really remove a large chunk of DNA and that can encompass many genes, are associated with multiple phenotypes. And so the inference we made is that in this subset of patients, because these genomic disorders are associated with kidney disease and both neuropsychiatric phenotype, the genomic disorders may actually be the reason why the patients have impaired neurocognition. So one way to look at this is to turn this, again, do a phenome-wide association study in an electronic health record system in which you have a lot of data about clinical health records. So it's an unbiased way of looking at it. So we wind up looking at the eMERGE data set that I was telling you about, 100,000 people who have genomic and electronic health record data. And we wind up seeing that, in fact, there's an association. So on the bottom, we have all the different phenotypes across all the different organ system and disease categories. And on the y-axis, you have the minus log p-value. And you could see that here above this red line are all the significant associations. So these genomic disorders in this data set are associated with renal phenotype, renal dialysis, but also a neuropsychiatric phenotype. And there are a lot of also nominally significant association with these types of different disorders. Certainly, they'll tell you that these genomic disorders appear to impair both neurocognition and kidney disease. It's not probably because of the kidney disease that we're now dealing with impaired neurocognition. And we've been able to now also replicate these findings in traditional data sets in observational studies. And one is the CKID study, in which we had actually neuropsychiatric tests that were done formally in the patients. And you could compare the patients who have one of these genomic disorders with those that don't have one. And there's essentially a 10-point difference in IQ between the two different patients. These are all patients with kidney CKD. And similarly, in the CRIC data set, which has about 3,000 patients with chronic kidney disease, adults now, 1% of them have one of these genomic disorders, and there's a big difference in their mini-mental status exam in terms of their scores, and also the level of education that's achieved in patients who have with these genomic disorders, and much likely to have gone on to further studies after completing high school education. Another example of this, or looking at the electronic health records in this large data set, looking to get insight into the pathogenesis of hypertension. This study was published a couple of years ago, looking at over 300,000 individuals with combining electronic health record data. And this identified 240 new loci for hypertension. And so these are illustrated here. These are the odds ratios that are associated with each one of these SNPs. I'm not going to go through the details here, but what's really interesting is that they wanted to understand a little bit of the biology associated with these diseases. And so they said, well, these SNPs code are in regions that there are genes. Where are these genes expressed? Because that'll tell us something about the pathogenesis of hypertension. And so they gleaned very little information from this because there was very little, data that we're able to get, or little enrichment for particular tissue types, except for maybe some vascular tissue that you could see tibial artery and aorta came out. And the reason for this was not because the studies were wrong, it's just that they were cross-referencing it with insufficient omics data. And so the data that they were cross-referencing to lacked kidney tissue gene expression. And so once this type of data became really available, and in the past year, we've had now 3 studies that have come out, which have performed multi-omics analysis, so especially looking at the kidney and gene expression. They've been able to now map these loci that have been previously identified in GWASs to specific kidney segments. And now, as a result of this, really explain what might be going on mechanistically through these genetic associations and come up with potential treatments. And I would encourage you to read these papers because they are really, really interesting. So I would summarize that genomic registries provide an opportunity for clinical genomics research. They enable discovery for clinical and genomic associations across many different traits. And the combined analysis of genomic and EHR data can clarify causality links between complex disorders. And then if you add now an overlay additional omics data on top of it, then you're going to try to get insight into disease pathways and point to potential therapies. So I wanted to thank a lot of my collaborators. This has been a multidisciplinary effort for the work that we've been doing so far, and thank all of our funders for sponsoring our research. Thank you very much.